I shall tell you what, in the course of my talk, what I mean by mining metabolites. Then I will briefly dwell on what is India's record in mining metabolites, and also discuss towards the latter part of the talk on what are some of the important questions when you are mining metabolites, such as why to mine metabolites, what to mine, where to mine, and how to mine. And finally, some take home messages for some of you who may be more interested in going a little deeper into this subject matter of this talk. So, let me straight away give some examples just to sort of highlight to you what I mean by mining metabolites. Now, here is a lady whose story I'll tell you in a minute. And but I paraphrase the slide by asking, how many of you would like to win an Olympic gold medal? Surely enough, I would say everyone who would be asked this question will say, yes, we would like to, I would like to win an Olympic gold medal. Now, to actually win an Olympic gold medal, I think you have to go and not meet me, but we have to meet this lady whose name is Naoka Takahashi. And by the name, you know it, she is a Japanese athlete. Now, Naoka Takahashi won the Sydney Olympic gold medal for marathon, and also she broke the record at that time for the women's marathon record. And when she won, the entire world was surprised because it was for the first time that a non-black lady went on to win the Olympic gold medal. Now, previous to that, the people who would win the gold medal were, typically Kenyans, Ugandan, Jamaicans, and so on and so forth. But when this Japanese lady won, they thought that there is some foul play. And so she was tested for dope. And she, of course, fortunately for her, turned negative for the dope test, indicating that she was not training on any anabolic steroids. But when persisted with this question about how she could manage to win the Olympic gold medal, her coach told that Naoka used to train on the juice of this insect that you see. Let me put the highlighter on this insect that is a wasp species named Vespa mandarina. So all that they do is they'll grind this insect up and make some sort of a juice like we make fruit juice and then drink. Apparently, the belief was that if you drink the juice of this insect, it gives you stamina because of the fact that this insect can fly non-stop for 75 kilometers. And so later research showed actually this insect is capable of doing that by having a very novel mechanism of burning its fat. Now, we'll not go to the fatty acid metabolism where you now break down fat to give energy. But all the same, this sort of insect extract was believed to enable human also to change their metabolism. Now, in human, I would like to mention here that we all store fat in our tissues and we rarely make use of it because most often our immediate source of energy is glycogen. Only after all of the glycogen is removed and oxidized, our body would allow us to metabolize the fat. And that is the reason why we keep adding fat in our body. And as we age, we become more and more fatty in terms of the amount of fat deposited. In the case of the wasp, fortunately for the wasp, that's not the case. So here, what happens is the wasp, despite burning its glucose, the sugar that moves around, and the trehalose that moves around, is also able to simultaneously catabolize fat. So there is no sequential sort of oxidation. So this is probably what was the gain from this insect. And later research showed, in fact, that is the situation. And so when athletes, the Japanese athletes went on to now feed on the base on this insect juice, apparently the molecules that are there in the extract enable the fatty acid metabolism in the muscles of the athletes who were running this marathon race. And it is probably because of that you had Naoka Takahashi winning the Olympic gold medal for the first time. It is today well known that in Japan, any athlete who is competing in the National Athletic Championship is made to go through a drink. International competition is made to go through a drink of this wasp, and thereby increase or improve the chances of winning the international competition. So some of the story can be picked up in Google if you want to know more about this one. I'll summarize the story in a minute after wrapping up a couple of more examples. Now let us take another example. Remember, we are talking about metabolites, some metabolites. So the other example is about a frog. Now every one of you may have held a frog in a hand at some time or the other. And you would know that a frog is very mucus, that is very slimy and all that. And in Chinese traditional medicine, the knowledge about frogs able to cure dog bites and then topical infections in the skin is well known. In California, there is a company, there was a company, now it is shut down. It's called Magainin Technologies. And this word Magainin comes from the word from Hebrew. This is a Hebrew word, which is equivalent to shield. So there it is believed that the frog skin is a shield that does not allow any infection to set in the frog. And so what they found is the frog skin secretions are loaded with lots and lots of antibiotics. And these antibiotics are the ones that prevent the frog from getting any bacterial infection, despite of the fact that the frogs live in places which are moist, which are having a lot of sludge and all that sort of stuff, which are in fact thriving with otherwise bacteria. So despite of that, the frogs do not get any topical infection from those bacterial flotsam. So this entire bunch of peptides that are secreted by the frog skin are related to the bradykinin peptides, and they have been shown to have a large amount of antibacterial activity. And the way to extract this metabolite from the frog skin is normally what people do is, to the thigh of the frog, they give an electrical stimuli, and that allows the frog to now respond by giving secretions of this peptide. And this company, Magainin Technology, which was located in California, what it would do is they would now do this frog farming. So they will every day morning, they will shave off the mucus secretion and then crystallize the protein, and that would be used as topical application for skin abrasions or skin infections and any other topical sort of infection. So this is my second example. Now let me go to a third example. Now all of you know about the leeches. Many of you may have been bitten by a leech at one time or the other when you had gone into the forest. And based on the medicinal property of the leech, there is a leech which is now Latinized called Hirido medicinalis, obviously because of the fact that this leech also was found in use in olden days, where if there was a topical infection of any sort, an abscess, the doctor would now, instead of incising the wound and cleaning the wound, would apply a leech to that part of the arm or part of the body where the abscess was located. And the leech would now suck out the blood, along with that, of course, the pus and all that. And while sucking the blood, the leech would also inject a anticoagulant so that there is a free flow of blood from the site of the wound. This became later a discovery which allowed people to now use this as what you may call as the blood thinner, the modern day blood thinner. In fact, hirudin, the compound that was extracted that is responsible for anticoagulation, was used in cardiac treatment for a long time before the advent of more, I would say, well-known medicines like aspirin and others and other anticoagulators. So this is yet another example. I'm giving you some examples to tell you basically how people have gone about extracting something from nature, whether it is in this case leech or a frog or whatever, to harness the metabolites that are embedded in them. Take, for example, an example from tree species. This is a well-known Cinchona tree, or also called the quinine tree, from Peru. And this tree, the bark of the tree is or contains this well-known alkaloid called quinine, which was discovered to cure malarial fevers. And in fact, it went on to become one of the most important metabolite that in some way changed the course of history, geopolitical history of the world, because the British apparently were able to conquer the rainforests of equatorial rainforests of Africa, the Indo-Malaya, not by their gunpowder, but by first making the soldiers say free of the malarial laden mosquitoes, because they would now go into the equatorial rainforests of Africa and the Indo-Malayan swamp forest by taking a shot of quinine called the gin tonic, a very bitter compound which was laced with quinine. So quinine, in fact, is said quinine has saved many more life than all of the world wars have killed together. So this is just one single metabolite coming from the bark of this tree. So I will now I'll just end my series of example with this celebrated work by Selman Waxman, for which he won the Nobel Prize, the discovery of streptomycin. Many of you will know about the discovery. I will not go into the details of that, but basically what he did was to now screen soil samples, thousands of soil samples, and from one of them discovered a very clear zone of inhibition, and found that in this case the zone of inhibition is due to the antibiotic called streptomycin against a very important bacteria that causes tuberculosis. So I'll skip these examples because I thought I'll spend my time a little bit in a different context. Maybe this I will not dwell now. So what is the take home from all of these stories? One is, I certainly feel that these 3-4 stories that I've told are very historical, in the sense that they are classical stories. So we all feel nice knowing these stories. I feel, for one, we feel excited listening to these stories. I believe all of you are teachers, and so it's very important that in our teaching line, we should also now evoke the excitement in the students that we now teach. And of course, if you are excited about something, you should be able to run home and tell these stories to your family, and the students should be able to share their stories with their family. But on a more serious note, what is common to these examples? What is common to all of these 3-4 examples, 4 or 5 examples that I gave you? The common thing is, in all the 4-5 stories that I just now spoke, if you now recall, just let us pause for a minute and recall the stories. The first story I told you is from the insect, where the insect was able to enable Naoka Takahashi to run the Olympic gold, marathon, and win the Olympic gold. The second story was regarding the frog, the bradykinin related proteins that act as antibacterial compounds. The third story that I told you is from the leech, which gives you the anticoagulator or anti-blood clotting factor. The fourth story I told you is about the Cinchona tree, from where we got quinine, which helped save the world from malaria. And of course, the fifth story was the discovery of antibiotics, the proper antibiotics that we today know. And one of them that I flashed is the streptomycin. Now, all of those examples, 5 examples, are or can be summarized by saying that they are products of mining natural resources. That is, you have mined some natural resource, whether it's a bacteria, leech, frog, insect, tree, or whatever. And so mining these natural resources for the products. What are the products? Here we are talking of the products which are basically compounds, chemical compounds. So I will synonymously use that chemical compound with that of metabolites. So we are now mining metabolites from natural resources. And this process of mining metabolites from natural resources is often referred to as bioprospecting. That is where we now prospect, that is, we now search the biological resources for some end use, that is your metabolites. In this case, it could be something else in some other case. But generally, in this talk, I will now relate to this metabolites. You can, of course, say bioprospecting as a definition. You can refer to it as a systematic search for new resources like genes, proteins, chemical compounds, and including whole microorganisms and other products that may have potential economic value to, of course, humankind. Now, all of you probably also, I would like to introduce another word here. There's a more frequently used word these days called bioeconomy. So one of the cornerstones of bioeconomy is bioprospecting, where you can now add value to your economy by discovering newer genes, newer proteins, newer compounds, newer functional activities from whatever organisms are there in nature. So that way you improve the bioeconomy as opposed to non-biological economy, which is mostly industrial economy. So, so the first part of my story was to give you an introduction about, in a very gross scale, of what you mean by mining metabolites. Now, before we go further, so the mining metabolites is something very similar to mining, let us say, mineral resources. Like you want to mine aluminium, or you want to mine bauxite ores for aluminium, you want to mine gold, you want to mine mercury, and so all of that. Or you might use it synonymously or analogously with mining for petroleum resources, or mining for, let us say, deep sea ores. So here I'm using the word mining with reference to mining the chemistries of organisms, the chemical compounds that occur in different organisms, which of course have a certain value. Just as mining fossil fuels has a value to you and me, mining chemicals may have a value to you and me in terms of its application in pharma industry, in agriculture industry, in general sort of human welfare. Okay, now let us come to the second part of my discussion. What is India's contribution to mining metabolites? Now, if I can ask you, what is India's contribution to mining petroleum resources? What is India's contribution to mining gold resources? What is India's contribution to mining coal resources? Now, we have some information in each of them, isn't it? Coal, we are one of the largest or major producers of coal in the world. Gold, we are, of course, one of the leading countries producing gold or discovering gold. And you can mine gold. We don't sort of make gold, we discover new sources of gold and mine those ones. We, of course, don't make coal, we mine the coal. So similarly, if I ask a question, what is India's contribution to mining metabolites? I would like to say this. Unfortunately, if you look at the track record, it is quite dismal. Unlike our country being known for coal resources, mining gold, mining petroleum, and all that stuff, when it comes to mining chemical compounds, it appears a little, the track is not very great. And whatever it is, if you extend the entire gamut of mining biological organisms, from genes, metabolites, pharmaceutically important bioactive molecules, biologically important properties of organisms, even whole organisms like microorganisms, we find that there are very few leads coming out of India. And that is a very sorry state to be. And because of that, if you look at the international market or global market for products from natural resources, it appears that our share of the biological capital out there is very, very less. We have not been able to harness the potential value of the biological resources that we have got. So we are a poor country with reference to as far as mining of metabolites from biological resources are concerned. And that is reflected if you now look at worldwide ownership of patents, contribution from India is very poor. So let me now dwell a little bit more at length on mining metabolites, and I shall restrict my discussion to the first three questions: why, what, and where, and leave the question of how to another lecture in the next couple of days. So I will talk about why should we mine metabolites, what sort of metabolites should we mine, and where should we mine. And we can have a discussion after this. So let us take the question, why should we mine metabolites? Now, if I were interacting with you in the class, I would straight away ask this question and generate some discussion. Unfortunately, in the Zoom class, that sort of interaction is not very convenient. So I'll go ahead and mention some points of what is the justification that why should we mine? Just because I'm taking the class, should we mine? No. As a country, should we be mining? Maybe yes. Let us see how. And I'll relate to it some recent sort of history also. Why mine metabolites? One is for sustaining our own demands. Now, this is the blue statements are a little old. I have not revised the slide, but it is probably true even today if this statements were to be updated. If you now take the top best-selling pharma products in the world, 80% of those, or even I would say between 60 to 80% of those, are purely derived from natural products. That is, those drugs are some sort of mimic derivative of some natural product compound existing in a plant, most often in a plant, or a bacteria or insect or thereabouts. Now, in our country, if you go to any pharmacy and you look at those compounds, you'll find that all of those compounds are coming from some other country. So we are a major importer of many of those compounds. Now the question is, had we discovered our own sources of the metabolites and discovered the use of those metabolites, we probably would not need to import them. So one of the strong justification why we should mine our own metabolites is to sustain our own growing demands. I hope all of you will agree with that. Second, there is this in this global market, unless you are competitive in the global scenario, you are going to be sidelined in the Committee of Nations. So to be globally competitive, I think India has to up its discovery, just like in any other field, but in this case, up its discovery in terms of metabolite discovery, and of course the properties of the metabolite, so that it can develop a market around those discoveries and become competitive globally. Third one is to circumvent intellectual property rights. Now, some of you would have read the famous intellectual property right conflict that happened over basmati, over curcumin, and few other. And you now know that unless our research base is strong, our discovery programs are strong, we often may lose out the intellectual property rights to some other third country, and we will not be able to stake claim on that simply because we would have lost the precedence to some other country. And so if we were to discover and mine our own metabolites in that area, we can now be strong in staking our own IPRs. Lastly, but I would say in some way it is going to be probably, if I were to do the slide again, it will be on the top of my slide. I think its mining metabolites also will generate a lot of knowledge that would now lead to lots of basic discoveries. I don't have the time to go into that, but then I can now, during discussion, I can tell you how that can be justified. By now understanding the, let's say, the antimicrobial peptides produced by an ant, or by understanding what is the sort of compounds in the trichome of a Solanaceae plant, one can generate a lot of basic knowledge that might have one day far reaching implication. So it is important to mine metabolites even for that. Now, for all of these reasons, and there are many more reasons I have not listed here, we could have discussed that, it's very important as a country that we should mine our metabolites. Just like as a country, we should be self-sufficient with respect to our food production. And so we should be as a country self-sufficient with respect to the chemistries that we now put to use for mankind. I will reflect on this again, this question about why to mine, with a very serious incident that happened in 2020, thanks to the COVID situation, and I'll come to that in a little while. Before that, let me tell you a story. The story is about the swine flu that occurred in 2009 as a pandemic. Now, all of us are very used to the word pandemic, and we just are coming out, hopefully, out of this pandemic. But in 2009, there was this swine flu pandemic. Now, this swine flu pandemic had a very striking similarity to what we are going through. Everyone was masked, and let us say, even in flights, the very few people who would travel in flights were also masked, and the airfares were very, very cheap, simply because no one was willing to fly, all due to the fear that you would contract swine flu if you were traveling with co-passengers. So in this year 2009, like we are scrambling for some cure for the coronavirus, people were scrambling for some cure for the swine flu. And thankfully, they had a drug called Tamiflu to treat the swine flu. And if you recall those days, at least the older or people in the audience would probably recall, there was scramble for Tamiflu, and people started holding Tamiflu should they get swine flu. Now, that is not the subject matter of what I'm going to say the next 2-3 slides. The subject matter is about the production of Tamiflu. Now, Tamiflu is derived by a 12-step derivatization process from a very basic primary metabolite called shikimic acid, which is present in all plants. Now, the shikimic acid, by the way, is the precursor for aromatic amino acid biosynthesis in plants. And so through the derivatization, which is from the basic material, that's shikimic acid, you get this compound, which is named commercially as Tamiflu. Now, so what happened in 2009 when countries world over wanted, including India, wanted Tamiflu, you had to generate the Tamiflu from this process. India had the know-how of this 12 step, and in Bangalore itself, there were 5 labs that knew how to make Tamiflu from shikimic acid, and therefore we can be self-sustained in terms of our dependence on our homemade Tamiflu. Unfortunately, the story is not as simple as that. The story becomes complicated when we now look for the starting material, shikimic acid. So our country did not have a source, or did not know of a source, naturally occurring source of shikimic acid, from where, using our knowledge, we could now do the semi-synthetic derivatization to lead to the production of Tamiflu. And so we were dependent wholly and solely, like many other countries, for the import of shikimic acid. And this happened to be from China. So China held the monopoly of supply of shikimic acid in the world because they had this species of plant called Elysium verum. This plant, commonly called as star anise, has in the seeds of its fruits, what you see here, these seeds are laden with shikimic acid to an extent of 2 to 6% by dry weight. So this was the supply or source of supply of the global shikimic acid market that China was running. So what happened when the demand for shikimic acid went up by many countries, including India? China imposed a moratorium in August 2009 on export of shikimic acid. So no private dealer in China can export shikimic acid outside of China without taking the Chinese government's permission. In other words, China seemed to hold at ransom the rest of the world in the supply of shikimic acid. Now, this is a story, and this story is as grim in 2009 as we are facing today, the story in terms of getting out of this coronavirus. But there is a twist in the tale. And what is the twist in the tale? In 2006, remember the pandemic of swine flu happened in 2009. In 2006, that is 3 years before that, there was another sort of disease, which was a very, very, I would say, quite a serious one, that was called bird flu. And even in the recent past, you would have heard of news of bird flu, which now wiped away a whole factory of this poultry business in many countries, including India. And the bird flu was also treated with Tamiflu. Actually, Tamiflu was discovered first for the bird flu and then later was extended to swine flu. So in 2006, we again had the capability of producing Tamiflu, but not having the precursor or this starting material shikimic acid. We were importing shikimic acid from China even in 2006, though the amounts of shikimic acid required at that time were not as high as was required in 2009. So in 2006, I was in DBT, the Department of Biotechnology, and one officer in the department asked me, Mark, can you people find out an alternative source of shikimic acid from Indian plants so that we don't have to be dependent upon China for the supply of shikimic acid? So why did this officer prompt me in with me this question? That day morning, there was a team from Taiwan that came to DBT, and they told that they are now just been successful in finding out an alternative source of shikimic acid. And Taiwan, the entire flora of Taiwan has around 400 or 500 plant species only, whereas we have about 18,000 plus higher plant species in our country. So the officer in DBT told me, look, when they can find an alternative source of shikimic acid from 4 or 500 plants, we certainly can find out one from 18,500 plant species. I told the officer, sir, your logic is not very correct. He asked me why. I told, look, Taiwan with a very small population wins so many gold medals in an Olympic event, whereas we with so many people don't win even one single gold medal. So the officer was beaten in his own logic. But I didn't want to push it any further. I told the officer, sir, we'll give it a try. And so they gave us a project. And from 2006 onwards, we started working on a project to find out alternative sources of the shikimic acid. So once again, just to give you a reference to what I'm talking about, shikimic acid is a metabolite. I want to mine that metabolite from Indian bioresources. So I want to discover which of Indian bioresources, that is plant, has a rich source of shikimic acid, which can therefore make us independent of the dependence, that is, it can make us free of dependence on China for the supply of shikimic acid. So what happened is, the rest is story. 3 years later, coincidentally in 2009, we published our first paper, whose title is Prospecting for Alternative Sources of Shikimic Acid, a Precursor of Tamiflu, a Bird Flu Drug. So when we sent this manuscript, swine flu had not yet become a pandemic. And so we have not used swine flu in the title, we have still used bird flu drug. So in this paper, we described our discovery and found 5 species. There are 2 species I've given the photograph of, Agathis and Calophyllum, and there are 3 more species. Each one of these species accumulates between 2 to 5% of shikimic acid in its leaves, while Agathis accumulates about 5% in its leaves. And this became the highest recorded source of shikimic acid in plant sources other than the star anise. And the advantage of these plant species over star anise was, in star anise, the shikimic acid is in the seeds, whereas this is in the leaf. So all of you would agree that the biomass being much, much more in the leaf could become more, in fact, a very attractive sort of source of shikimic acid compared to the seeds. So when this was published, and coincidentally, it was published on March 25th, 2009, and I think on April 1st or 2nd, the WHO declared swine flu as pandemic. So as soon as this was published, lot of Indian and international press got hold of this paper, and just 2 or 3 headlines I shall put here. The headline screaming said, India steps up search for rare ingredient of swine flu drug. Western Ghats hold key to flu fight. Indian plants offer new source for Tamiflu precursor, and so on and so forth. When all of this were being sort of put in the newspapers and lots of other media, international media, in August 2009, I told you China imposed moratorium. But slowly, when the proof of principle and all that, I don't want to tell you other stories about this one, went about, China silently withdrew the moratorium, because now it knew that applying a moratorium actually does not serve any purpose, because if it had continued with the moratorium, we would have exploited commercially on industrial scale the many sources of shikimic acid that we discovered from the Indian bioresources. So the moral of the story, besides the 4 points that I gave you in the beginning, is that forget cashing the biological capital, forget making money. To save our own skin, we need to mine our own metabolites. We need to bioprospect our own metabolites. We need to now find our own sources of medicine. We need to we need to make our own vaccine. We need to now make our own food. We cannot be dependent on a third country to now supply us in the time of need. So that is probably one of the most powerful justification to bioprospect our resources for the value that they have got. Okay, so second question is what to mine. Now this is, as students of science, it's a very, very wide sort of question. There's no bandwidth for this question, what to mine. It can be anything and everything, as I listed in the earlier slide. Let's take the case of genes. Now, all of us have heard of BT cotton, BT this, BT that, and all of that depends on only one gene, that is a BT gene. But if I ask you how many new genes have you used in transformation, in the development of transgenics from Indian discovered or India discovered genes, I would say not more than 3 or 4 we might have discovered that may have commercial implications. Similarly, if you now talk about metabolites, we have not made great discoveries. For pharmaceutically important biomolecules, not much, and the list goes on. So what to mine, though my emphasis here is on metabolites, You can mine a whole range of products from biological organisms. It depends on one's interest, one's training, and one's inclination to go about. But any one of them are justified. Take, for example, the insect kingdom. One can now mine beetles or other insects for whatever properties. One can mine frogs for their antimicrobial peptides. These are two gentlemen who have been interested in frogs. Dr. Santosh Kumar from Rajiv Gandhi Institute of Biotechnology and Dr. Biju from Delhi University, who specialists in describing frogs, discovering new species of frog. So we have the forces that can now lead up to great discoveries. The last question that I'd like to deal now is where to mine. So we have asked why to mine, what to mine, where to mine. So where? Of course, you are going to mine in your lab or whatever. But what I'm saying is, what is the source of the biological organism that you are now taking? Let me give you one or two examples. There's a most important, I would say, good illustration that all of you will know about the discovery of the thermophilic bacteria, thermus, from the sort of hot springs, and which became later the source of the heat-resistant enzyme Taq polymerase, which I'm sure many of us would have used in our lab. And obviously, before this bacteria could be discovered, there was a certain logic of asking which sort of place should I now discover a thermus species from? Surely not the cold spring, surely not some other mesophytic place, but a hot spring. So you are now in some way rationalizing your mining of organisms from habitats that may predispose them to be adapted to some adversity, like in this case, the high temperature. But sometimes, unlike the discovery of thermus, you cannot always sort of say that deep rationalization might work. Take an extreme example of the Easter Island, which is given in this box, which actually is a place probably far away from any human settlement in any of the other continents. And this is totally uninhabited. And there's a whole bunch of stories about the Easter Island and the iconic monoliths that you see on the island. There's a whole bunch of story that some of you may want to might want to learn about, read about. But what I would like to focus my attention on is on Indian chemist called Suren Sehgal, who emigrated from India to Canada. And he was a chemist and worked in a private company in Canada. And the Canadian company was interested in bioprospecting all sorts of strange places for bacteria and actinomyces, fungi, and this and that and all that. So Suren Sehgal went to Easter Islands. Remember, Easter Island is barren. There are no trees on Easter Island, and of all the places, I would not have elected to go to Easter Island for bioprospecting. It's a different matter that maybe one would have gone there as an exotic sort of place. But Suren went there and collected soil samples from there. And in the soil sample that he processed after collecting and getting back to Canada, he discovered an actinomyces called Streptomyces hygroscopicus, and found that one of the strains of this actinomyces produces this large-looking molecule, what you see here, that's called rapamycin. And since it was a private company, the company was interested to find out the properties of rapamycin. And shortly enough, Suren found that this compound has a very strong antifungal activity. So antifungal activity. So it was sort of used as an antifungal compound for some time, and then kept in the shelf of this company until years later, Someone else tripped upon rapamycin and found that rapamycin had another wonderful property, and that was it was one of the then known strongest immune suppressant compound. Soon enough, after knowing that it has an immune suppressing property, it became to be the choice of medicine for immune suppressant administered to any transplant patients. In fact, thousands of kidney transplant patients, if they have extended their life, it was due to rapamycin, which helped them retain the kidney donated by a donor and not rejected by their own immune system. And that work was done by rapamycin. And so you see here the discovery that Surin did from Easter Island went up to make a profound impact on the human health of lots and lots of people that had to retain the transplant. And this comes from a streptomyces. We would never have expected this even in our vaguest dreams, that we would get such a wonderful discovery from the barren, rocky, not rocky, sorry, barren Easter Island uh, out of nowhere. Okay, so where to mine? In the general context, Easter Island could be an exception. This graph tells us just a simple algorithm of where to mine. If I were to draw on the x-axis the biological space, that is biological diversity or space, and on my y-axis the chemical space or the chemical diversity, all of you would agree with me that chemical diversity will scale in some increasing way, either linear or in a curvilinear manner, with increasing biological diversity. That is, more and more biological diversity you harness, you are likely to now increase the chemical diversity or metabolite diversity that you will accrue. With this simple sort of logic in mind, you would now go and ask where to mine. If you ask that, I would say generally in areas or sites where there is rich biodiversity. And that is where your chances of hitting greater chemical diversity and unique and novel molecules will be higher. So this is a map of the world where the pink or purple, sorry, purple, deep purple and light purple sort of polygons all over the world depicts to you the sites of very high vascular plant richness. And these incidentally are the sort of hotspots of biodiversity in the world, which spans about 10 to 15 degrees north and 10 to 15 degrees south of equator. And so these are the places that you would be better off in visiting and collecting your samples, either plants or insects or whatever, to maximize the probability of hits to make whatever the discoveries. Incidentally, those are the very same sites that are also the seats of older civilizations. And so you would agree with me that these are the sites that would also offer us a rich traditional knowledge systems about the plants and other organisms around the homesteads that people have lived for years on end, and therefore we can have a better launch into discovery from such places rather than from places that had not been inhabited by humankind. So in summary, therefore, if you now come to our own biodiversity, that is the Western Ghats and the Northeast India, which are sites of huge amount of richness. If you now look at the Western Ghat, which is one of the 4 biodiversity hotspots in the country, this area, this green patch, which is the Western Ghat, constitutes about 5% of our country's geographical area, but has about 25% of all of India's biodiversity. And so, therefore, you have an excessively rich diversity packed in a small area. And so the probability of discovering anything here might be much, much larger than if you were to do, let's say, in the Deccan Plateau. And indeed, there have been lots and lots of expeditions to the Western Ghats by the colonial forces, the Portugal, the English, the Dutch, and then their discovery was more not on metabolites as much as the whole sort of products. In this case, black pepper, and there's a very nice story about that. But because time is running short, I'll not go into this story. I'll sort of skip all of this and give you this one table to show. Uh, Author called Pushpam Kumar went on to value the biodiversity or hotspots of the world, which are listed here, and found that by the endemicity, uniqueness of the flora that you have in the different hotspots, he ranked the Western Ghats of India as the fifth most valuable hotspots in the world with respect to its biodiversity, bioprospecting value. So, if you take a hectare of forest in Western Ghats compared to a hectare of forest in maybe Californian floristic province or whatever, you would find that the bioprospecting potential of Western Ghats will be that much higher because of the high amount of endemicity of the flora in this area. And so you can be guided by a whole number of rationale, but the blanket rationale would be go to place where there's a high biodiversity. You probably are going to be better off in discovering whatever you have chosen to discover. How to mine, as I mentioned, I'll talk to you about this how to mine exclusively as a separate lecture. Maybe in the next lecture when we meet, I think it is on 20th or whatever, and then we can now continue the discussion there. Okay, so what are the summary and what are the recent initiatives in India? Now, I'd like to summarize with this slide a little bit of generalization. And again, as teachers, I think this message might be well borne. This is a story that an anecdote of Sir C.V. Raman and his diamond and the diamond ring. So the story goes something like this: that Sir C.V. Raman once went to Madras, then Madras, where his nephew had recently got married, and also the nephew was a physics lecturer in the Madras Presidency College. So when Raman went to the nephew's house, like any newlywed Indian bridegroom, this nephew was sporting a diamond ring on his finger. And so Raman, when he saw the diamond ring, rebuked thus. He said, "I say, why are you wasting the diamond ring on your hand?" Nephew was a little taken aback because this is not the politest thing you would tell when you visit your nephew, that too just after he got married. So Raman had his coffee and left. And long after, this nephew was wondering, why did my uncle tell thus? And it took him a few days to realize that the import of this message probably was something else. And so what this guy did, the nephew, being a physics lecturer, he thought, oh, maybe Raman did not want me to sport the diamond ring on my finger as much as he probably would have wished that I put this diamond ring to study the light scattering by diamond. And so what he did, he took the diamond ring off his finger and did an experiment to study what is the sort of light scattering signature of the diamond, and then published that paper. Incidentally, that was the first paper on light scattering by diamond, and sent the paper to Bangalore to Raman, and Raman looked at it, and then after that he sent a small message once again back to the nephew. What was the message? The message was, "Now you have put to good use your father-in-law's gift." Why am I telling this story? C.V. Raman's quip can be extended to our own biodiversity, which I would refer to our own green diamonds. What has happened in our country is that we have prided ourselves on the rich bioresources of the country time and again. We have also prided ourselves on the rich indigenous traditional knowledge that our grandmothers, our people have. But we have done very little to put the resources to good national use. This is something like sporting the diamond on our back or on the finger and not doing anything about it. Besides just, I would say, gloating that we have this richness, we have such rich ITK, but we have not done anything to harness those richnesses. So, as someone mentioned, it is time that we stop bloating of our rich biodiversity and put them to good national use. So I believe as a country, amongst the many other focus of attention that we have are paying, I think mining metabolites should become a very important activity from the bioresources that can enable us to power the bioeconomy of this country. So the Department of Biotechnology in the last 15-20 years has come about with major programs on bioprospecting of all sorts of organisms, on insect resources, micro amphibian resources, mining genes, and a whole bunch of things. And I believe some of you might already be beneficiaries of this program. If not, I think you should now open up the DBT page and see where you fit in and how you can contribute in your own way, nurturing your own interest. And in the passing, if it has some implications, it is well and good. So I'll stop here. Thank you very much, Dr. Khan, and thanks to all the participants for listening in. And I believe we have about 10 minutes. If we need to have interaction, I'll switch on my video now. I will I quit sharing my screen so that we can have a chat. Thank you very much.
Thank you, sir. We have one question.
Yeah.
Can we analyze traditional use of plants for bioprospecting?
Can we?
Can we analyze traditional use of plants for bioprospecting?
I don't know if I understand the question correctly, but can you ask the gentleman to ask me the question?
Pradeep sir, can you unmute him first?
Sir, good morning, sir. Professor Chari, good morning. Nice to see you again. Thank you very much.
Sir.
Yeah.
Sir, I want to know that in India, there are so many plants used by the tribals as their medicines.
Yeah.
So can those plants be bioprospect?
Yes, please. As I told, in fact, you can use the traditional knowledge systems by the tribal groups or communities to get a lead and ask more questions that will lead to discoveries. I shall talk about this in my next lecture, where under the title of how to prospect, how to bioprospect, I have one section on how to use indigenous traditional knowledge systems to prospect metabolites. You're very right. We can do that.
Okay, sir. Thank you. Thank you.
Yeah, thank you so much.
Mujahid sir, can you ask question? You were.
It will be nice. Just what I would like to mention on behalf of Dr. Khan. See, in the Zoom meeting, our interactions are very limited, but I still feel we have to become adaptive because we have become used to the Zoom for the last 1 year now, that we should give 10-15 minutes so that any one of you, I don't believe in this writing in chat and all that, you simply unmute and then talk. I think that way some informality will come in the course of our discussion. That way it will enrich the participation of the entire group.
Mujahid sir, can you ask question now? He was trying to ask something.
Okay. I see Dr. Akhilesh Tyagi, Professor Akhilesh Tyagi on the screen, but I think he's muted and...
Yeah, he's in
Good morning, Uma. Good morning.
Good morning, sir. Good morning, sir.
I listened to the last part of your presentation. Nice.
Actually, I was telling them that, in the Zoom meetings, I think it's
I heard that. Yes.
So that way we can have more interaction.
Sure.
So participants, I would request you all to sort of, I won't say wake up, but become more active. That will allow little more sort of animated discussion and get the best out of the speakers and also from the entire discourse that we will have. Maybe from next lecture onwards, you can now do that. Or we can also give 20 minutes and then stop and then have 5-10 minutes of discussion, then again go like that. So something like that we should do.
Uma, can I ask a question?
Yeah, please ask.
Can I ask?
Sir.
Yeah. My question is that when we talk of utilizing bioresources,
yes, sir.
In India, how we will be able to approach in a sustainable manner?
Very good question.
That our resources are maintained and we are still able to get benefit to the society that we desire.
Yeah, very good question. This is a question which is on expected lines, and this often asked. So the answer to this is quickly twofold. One is, by knowing what are the hidden sort of properties or hidden virtues of a plant, you now automatically try to conserve the plant. And if you did not know, you of course out of ignorance would not conserve the plant. That is number one. Number two, having known that what is the richness embedded in a plant, there is a fear that one would now indiscriminately extract the plant, and then as Professor Tyagi is probably prompting, that we may now drive the plant to becoming vulnerable or eventually also extinct. But many of the, I would say, historical examples show that in very few cases only that has happened, but in most cases actually it has led to a sustainable management of the resources, maybe by domesticating the resource out of the wilderness, maybe by pulling out the genes, maybe by doing a synthetic biology of or synthetic chemistry of the metabolites that one is interested in. The best example could be artemisinin that comes from the plant Artemisia annua. Another example, our own example of camptothecin is where we have now started a semi-domestication of a plant that was growing in wild, which we found to be a highest producer of camptothecin. So there are many ways to now go about doing a sustainable management of the resource that you have found use of, or where there's an importance to be harnessed. That's the answer,
Thank you,
sir.
Thank you.
Yeah. So if there are no other questions, maybe I will.
Yeah.
Khan?
Dr. Alim Khan?
Sir, yes, sir.
On behalf of the academies and on behalf of Telangana University, on behalf of Dr. Alim Khan and Refresher course participants, and on my own behalf, I thank Honorable and respected Professor Uma Shankar sir for having given us an opportunity to deliver this particular very innovative and knowledgeable and informative and mostly liked lecture. And in order to preserve all our biodiversity and ex situ and in situ conservation, which needs to be taken up, and legal protection is also needed for this particular one. He spoke on various aspects, and I'm grateful to you, sir, Professor Shankar, sir, for
Thank you.
having given us an opportunity. And you are such a kind of Dayan in biology, which you deserve all our respects and regards. Thank you very much, sir. Thank you very much on behalf of the academies and on behalf of Telangana University. Thank you, sir.
Thank you, sir. Thank you.
Thank you very much, sir.
Thank you. Thank you.
We have 3 more lectures again back. So we will be again seeing sir back. So now, dear participants and our course director, very good morning, one and all.
First, may I say my regards to Professor Chari and also
Sir,
to
sir.
Dr. Khan.
Sir,
Namaskar.
namaskar, sir. Namaskar, sir. Namaskar, sir.
Sure. So it is proud moment for Department of Botany, Telangana University, and the Refresh course team to have us today with us, Professor Akhilesh Kumar Tyagi, sir. He is from University of Delhi, and earlier he has served as director for National Institute of Plant Genome Research. He led Indian initiative on genome-wide sequencing in rice tomato and desi chickpea, heralding the era of high-throughput genomics in India. He made pioneering contributions to the area of neo-sub-functionalization in gene families during evolution and gene function during water deficit response and grain development in rice. Tyagi sir has received J.C. Bose National Fellowship Award, National Bioscience Award, National Academy Science of India Reliance Industries Platinum Jubilee Award are among other awards to his credit. He is a fellow of National Science Academies and the World Academy of Sciences and had the distinction of serving as President, National Academy of Sciences, India. So with these few words, I would like to request sir to start sharing his screen. Over to Tyagi sir.
So you will allow me to share now?
Yes, sir, you can share.
Your co-host, you can start sharing.
Okay, all right. So are you able to see me on the screen and listen to me, my voice?
Yeah, your voice is there and your screen is also there, but your camera is off.
Okay, camera I can put on if you want that. I was trying to save some bandwidth. That's it.
Is it so?
All right. So you allow me to begin now?
Yes,
Yes,
sir.
please.
Sure.
All right. So thank you very much for giving me this opportunity to interact with your faculty and students and all participants in your meeting, which is supported by all the 3 academies. So it is a unique opportunity to thank them and also the organizers of this meeting. And as the title shows, I would be talking about diversifying crop genome. Now, of course, very first question comes, why diversify crop genome? Now, as was said in the earlier presentation, the diversification is largely to sustain humanity and also to sustain our planet in a way that we can live for much longer than what is expected because of all these activities that we have. So sustainability is the issue in this case, and that is the reason the United Nations has set 17 sustainable developmental goals for the next 10 years or so. And in this, you will see the goal for number 1, no poverty, 0 hunger, good health and well-being, affordable clean energy, climate action, life below water, and life on land. These all connect with agriculture in one or other way. And agriculture in turn depends on the live organisms, and therefore agriculture scientists and geneticists in particular have been busy in improving the organisms by way of diversifying the genome. But it is not only the scientists. Nature has also been doing this diversification over the time, which has sustained the evolution also. And you can see in this tree of life that how bacteria, archaea, and eukaryotes have come into existence by vertical gene transfer. And of course, any change which took place in those genomes was transferred to the progenies. But in addition to that, there have been horizontal transfers also. As you will see these vertical lines, the major ones are the creation of mitochondria and the plastids in a eukaryotic cell. And in addition to that, there are relics that many times there have been horizontal gene transfer as well. So vertical as well as horizontal gene transfer has been the nature's instrument to change the genome. We have, as human beings, also modified the plants over a period of time. For example, selective breeding has been done to improve the crops. Cross-pollinations between diverse varieties and sometimes even in species have been attempted. And hybridization has been taken over as on experimental basis. This all has resulted in genetic modification, and it is exemplified in this picture by way of the progenitor of the present-day maize in comparison to teosinte, from which the maize seems to have emanated. And this all was possible because of the selection and also the breeding. So there has been genetic modification going on for many, many years. Also, you can see in rice, for example, if you compare the wild rice with that of the domesticated rice, then you will find there is a great difference between two in their habitats and also in their phenotypes. For example, the wild rice is perennial, whereas cultivated rice is annual. The wild rice has shattering of the seeds, because of which seed loss is there, but the cultivated rice has non-shattering seed habit, so we can collect the seeds. The wild rice was prostrate in habit. The present-day rice is erect in habit. The wild rice had awns which are undesirable. Present-day rice largely lacks the awns. The color of rice was red. Now we have white, which we like. And of course, there are characters like outcrossing versus selfing and low grain yield versus high grain yield. So how this whole happened between wild rice and domesticated rice is because of the genetic modification and its selection. And therefore, this genetic modification or diversification of the genome has played a great role over the period of time, and many of these traits which are shown here are controlled by genes which we call as the domestication genes. Now, if we go to this and we try to show what we have done in relation to this, the picture shows that we have been participating in the international programs on genome sequencing. The first step was on the decoding of the rice genome, and about 400 megabases of this genome were sequenced by 10 nations. India worked on chromosome 1. Then came the next stage, and in this stage, tomato genome was sequenced, and we worked on chromosome 5. Now, between this and the tomato genome, the difference was that while the decoding of rice genome was done by capillary-based sequences, the tomato genome was a hybrid genome between the capillary-based sequencing and the next generation sequencing. And of course, then India has participated in sequencing of wheat also, which has been done in addition to our institutions, that is Delhi University and National Research Center for Biotechnology in ICR. In this case, PAU also participated. And that way, multiple institutions gain the capacity to sequence the entire genome. Once this was done, we had the next challenge, that can we sequence entire genome in India? And because of good human resource, we could sequence, as a matter of fact, several genomes within India. For example, in chickpea, desi chickpea, Kabuli chickpea, wild chickpea have been sequenced, pigeon pea genome has been sequenced, finger millet, pearl millet, tulsi, neem, mango, and dark jute. All these genomes have been sequenced subsequently, and this has been done by Indian scientists themselves. So India acquired the capacity to sequence the entire genomes and stands very well in the international scenario. Now, another thing that genome sequencing revealed relates to genome evolution. Now here I am taking an example from rice and its relatives, that how these chromosomes have evolved in these species. So we can start from this tree, and the very first thing that we see is the whole genome duplication about 60 million years ago. And this was the time when we had the dinosaurs disappearing, and there was a change in the climate, and probably that climate change resulted in the whole genome duplication. And then, of course, we have diversification between rice, wheat, sorghum, and brachypodium. But question comes that how these chromosomes got established. For example, it is considered that rice, which has got 12 chromosomes, it has a duplication, and because of which 5 plus 5 chromosomes were created. At the same time, translocation and fusion added 2 chromosomes, and therefore 12 chromosomes of rice came into existence. The question is, where is the progenitor of it? And when brachypodium was looked into, it was found it has got 5 chromosomes, and therefore one can consider it to be the progenitor of rice. But when its genome was looked into, it was found that no, its chromosomes also have gone undergone duplication. They have gained 2 chromosomes, and then lost 7 chromosomes, which brachypodium has got 5 chromosomes. Now, this information could be achieved because we can look at the chromosomes on the basis of the order of the genes. And order of the gene can define whether there is a duplication or there is a loss of certain genetic information. Anyway, we had another plant, which is sorghum. It has 10 chromosomes. So it was considered that this represents the rice genome in a duplicated step. But again, when this was looked into, it was found that there was duplication, gain of 2 chromosomes, and then loss of 2 chromosomes. So 10 chromosomes came into existence. In case of maize also, there are 10 chromosomes, but its origin is quite different from that of sorghum, because in this case, we not only have a duplication of chromosomes, but we have a tetraploidization, or 2 duplications have happened, because of which, as you see here, 5 plus 5 plus 2 minus 2, that is 10 chromosomes, then second duplication, then loss of 10 chromosomes, and that is how the 10 chromosomes of maize came to exist today. And of course, in case of wheat, there has been loss of 5 chromosomes, because of which the basic chromosome number is 7 in this case. So you see that by learning about the information of the genome, the very first thing we start to learn is about the origin of the genomes. And these genomes do not follow a simple path, but have gone through duplications, translocations, and fusions at different times to come into the shape in which they exist today. So that is how this relationship on the basis of the genes led to the understanding of the relationship on the basis of the chromosomes and about the origin of the chromosomes. In addition to this, it was reported that there are about 30,000 genes in case of rice. But even today, we know the function of only about 5,000 genes. And these genes have been worked out either by gene-tagged mutant analysis, by overexpression or knockdown transgenics, or by map-based cloning. So major three technologies have been used for this purpose: gene tagging, transgenics, and map-based cloning. And by doing this, we have found large number of the genes which represent growth and development, then biotic stress, and hormone signaling. Important thing to remember is that there are another 25,000 genes whose function we do not know as of today. And therefore, a lot of work remains to be worked out, to be done, when we will be able to see that we understand the function of the whole, all the genes. And don't forget that genes are only about 5% of the genome. There is, of course, repetitive DNA or selfish DNA. So function of that also remains to be worked out. Therefore, the area is open to a lot of discoveries and also has got a lot of challenges in itself. In any way, the next thing is that how do we start to utilize it to diversify the genome and also to improve it. So towards that end, we started to look for the expression profile of the genes, all 30,000 genes, which were there on a chip, were used for analysis of the expression during different organs. Say this slide is showing you that which genes are expressing in seed, and they belong to the category of transcription factors and signal transduction components. So you can see that these genes are expressing only in the seed stages, which we have selected, not in the panicles and not in the leaf, root, and seedling. So you can know by doing transcriptome analysis, the genes which are specific for any organ or for any state that is abiotic or biotic state. And you can very well infer that these genes which are specific to this particular state should have a function in that particular state. So you get the targets for further analysis by doing the basic transcriptome analysis in different conditions and also in different stages of development of the plant. For example, this is one of the target genes which we have worked out. The gene belongs to the mediator complex. Now, mediator complex is communicating between a regulatory transcription factor and RNA polymerase to control the expression of the gene. And we found by protein interactions that in the leaf and lateral organs, this mediator interacts with YAB5 transcription factor. In case of seed, it interacts with METS29 transcription factor. And in case of anther, it works with TDR factor. So the communication from these transcription factors goes through the mediator to that of the RNA polymerase, and expression of the gene in a specific organ takes place. And therefore, this gene becomes important for shaping the anther, seed, and lateral organ functions during the development of the rice. This approach, of course, is based on the transgenics. That means we have taken the gene, we have overexpressed it, or we have knocked it down by doing RNA interference transgenic development. So that is how we can know the function of any of the gene in rice or in any other plant by overexpression or knocking it down. Now, next stage, of course, is that to make use of this variability, which exists already in case of plants, and here we are showing the variability as it exists in relation to the panicle, in relation to roots, and also the seeds in the rice germplasm. You will be happy to know that India has about 100,000 accessions in NBPGR for rice, and a lot of variability is there which can be made use of. So we started to think in this line, and that point we use genome information and also the phenotyping together to reach what is called as marker assisted breeding. So in marker assisted breeding, you have two parents, for example, here the resistant parent and the susceptible. The resistant has got a marker DNA here, which is of a smaller size, whereas susceptible has got a marker band, which is of high molecular weight size. So we can cross these and get F1 and then make large population in F2. And then question is, how do we phenotype? How do we know that the gene has been transferred and the resistance has been gained? So in place of doing phenotyping of large number of the plants, we can do the genotype. And here you see that there are some plants in which there are two bands. That means they are heterozygous, whereas others are there in which there is only one band. Like this large size band represents a homozygote of susceptible nature, whereas this small size band represents a homozygote of resistant nature. So out of this population, you will be able to select the resistant plants even without phenotyping that. And that becomes the marker assisted breeding, which is a combination of genomic information with that of the breeding approaches and the phenotyping taken together. Now, doing this, a lot of work has been done in India by different laboratories. It is not necessary that we have to do everything. So I am giving you that picture. For example, submergence tolerance gene has been introduced into Indian varieties, and this is now being combined under a program which is called as gene permitting QTL2 variety, largely done by ICR institutions. And a drought resistance gene is being combined with that of submergence tolerance and also salt resistance. And here you see the performance of a drought resistance gene, and which also has got the flood tolerance gene involved in this. So that approach has been done. Also, the bacterial blight related genes have been permitted into Pusa basmati rice by genomics assisted breeding. Like here, 2XA genes, which provide resistance to bacteria, Xanthomonas, have been put together, and the resistant varieties have been developed by way of gene pyramiding. We also have the approach going on in which the bacterial blight will be combined with that of blast resistance and also with salinity tolerance. So these elite rice will also become available. I am told that already about 3 million hectares of rice is being grown in India, which has emanated from genomics assisted breeding. And so we can see that it has already been contributing to the bioeconomy of about 30,000 crore rupees. And in future, it is likely that many of the varieties would be developed by genomics assisted breeding and would cover larger areas of rice cultivation in India. Already work has also been started in case of rice land races on biotic stress like bacterial blight, blast, Magainin disease, false smut, sheath blight, brown plant hopper, and on abiotic stress, drought, salinity, submergence, and nutritional deficiency, and other traits related to that of grain quality. So when these land races are analyzed at genome level for this kind of traits, we would expect new QTLs and genes emerging from them. and then they will be bred into the cultivated varieties. Thus, farmer will be able to get the benefit of genome approaches directly in their field, and better seeds would be available for them, which can sustain the environment and also possibly provide higher yields. So that is the power of this first technology of genomics assisted breeding that I am telling you. The work is not restricted only to rice. In India, work is also done on maize, and nutritionally rich maize has been already released. This QPM variety, Pusa Vivek, has been released by another ICR institute. Also, in South Campus, we have a group which is working on Brassica genomics, and they have sequenced Brassica gensea and Brassica nigra genome. SNP data has been collected from 8 varieties of this. And then what has been done is that by using molecular markers, traits like glucinolate content, low erucic acid content, and white rust disease resistance has been achieved. And these things are also on the way to the field of the farmer. So other crops have also been taken up and are taking advantage of genomics assisted breeding. Now we also moved in this direction, and we established a panel of genotypes with about 500 and analyzed their genomes. And what we found in them was about 500,000 SNP, single nucleotide polymorphic markers. Now, these germplasms varied in the seed quality: length 5.6 to 14.5 millimeter, width 1.7 to 6.7 millimeter, and 1000 grain weight 9.8 to 48.7 grams per 1000 grains. Now, this kind of variability and this kind of genomic information was combined to ultimately find out the genes which are responsible for seed development. And we found, by doing a genome-wide association analysis, several of the SNPs in 31 genes which significantly associate with grain size. And we could not only in these 31 genes identify 10 genes which were earlier reported to be contributing to seed traits, but we also identified another 20 genes or QTLs which were providing or associating with that of the grain trait. These, of course, have been used to find out on the basis of genetic mapping that this association between the gene or QTL with that of the grain trait is really genuine. And so the genetic mapping was also done in the population derived from the 2 parents, one having the low grain weight and another having the high grain weight. And we did haplotyping to find out that which of the genes where alleles are responsible for grain weight. For example, here there is a gene for Matzbox transcription factor. If you see, there is a change of 1 nucleotide here. In place of CCT, it is CTT. Therefore, in one allele, serine is encoded. In another allele, phenylalanine is encoded. And if we see these haplotypes, we find that the serine one relates with low grain weight, whereas the phenylalanine one relates with that of the high grain weight. So it is possible ultimately to start with variability and genome information and ultimately to reach a gene or allele of the gene which will be able to provide high grain weight if this is used for transfer into the varieties. Similarly, in case of chickpea also work has been done, and here 2 varieties, one having a small seed size and high seed number, and another size and low seed number, they were crossed. And by crossing, we targeted a ABC transporter gene for transfer, and it was possible to get a improved desi variety which had 20% more yield 13% more productivity, and 15% more protein content. This has been approved through the national trials also for release in some parts of India. So this is another example that I am trying to give from the work which emanated on the basis of rice and the chickpea genome sequencing. Now we move further and we find that over the period of time, the sequencing has become cheaper. Now, as per Moore's law, the price should have declined on this graph, blue line, but price has declined much more over a period of time from 2001 to 2020, when many of the genomics programs have been undertaken. And this has given rise to sequencing approaches from gel-based sequencing to capillary sequencing to next generation sequencing and also to third generation sequencing. So combination of low cost and high throughput allows many more genomes to be sequenced, and even in one organism like rice, one may like to sequence thousands and thousands of the genomes so that variability is deciphered on the basis of single nucleotide variability that may exist in these varieties. Now, this brings us to a new concept, which is called as pangenome concept. Now, a pangenome refers to full complement of genes in a biological clade. For example, there might be a basic number of the genes which are present in one variety, but other variety may have some additional genes. And if we combine the genes of large number of the varieties, we are reaching the pangenome approach. For example, accession 1. Here it has got this C and this G, these C genes. These are present in all varieties, but each of the accession has got some genes which are different, like here, present in this variety, but not in this variety, and it is present in other varieties. So that is how the genome of different accessions of rice also shows variability as we sequence thousands of the accessions of rice. And then we can make use of this information again to reach to the QTLs and genes which are responsible for improving the traits of interest. Now I will give you an example what pangenome represents. For example, normally it has been proposed that there are about 30,000 genes in case of rice. But by doing the pangenome analysis, 12,000 new genes have been found. And this is true not only for Oryza sativa, but for other species also. So that is the power of analyzing pangenome. And some interesting QTLs which have been found by doing pangenome, which means they were not present in the variety which was sequenced first, but were present in other varieties, represent the submergence tolerance QTL, which was found to be present in the variety FR13A and IR4093126, not Nippon Bare, which was originally sequenced. Similarly, the QTL for phosphate utilization, PSTOL1, has been found to be present in Casselleth and not in case of Nippon Bare. So this shows the power of the extra genes which are present in varieties and represent the pangenome. We also used this information to find out the QTLs related with grain length, grain width, length to width ratio, and 1000 grain weight in case of rice. While we found the QTLs similar to those which are present in the first sequence genome of Nippon Bare, we could find in pseudochromosomes about 8 QTLs extra, which relate with that of the grain traits. And this shows the power of analyzing the extra genome also, that is the pangenome, and looking for useful genes in that particular area of the genome of rice. And of course, we could identify several of the potential candidate genes in these QTLs on which work is still going on. So what I'm trying to say is that we have genome and we have pangenome. We have, of course, the genome-wide association analysis, and we have genetic analysis and haplotyping. And by this, we are able to connect the gene with that or allele with that of the trait. And then this particular allele could be transferred into many more varieties which our people in India are eating. And they may get a seed, which is an improved seed for various traits which are desirable in case of rice. This is possible by way of the genomics. But what is for future? It is being thought that we should sequence the entire eukaryotic life on the earth. And this sequencing is being proposed through a project which is called as Earth Biogenome Project. Now, this is considered that if we have information about the genomes of all the eukaryotes, plants and animals, then we could probably revolutionize our understanding of biology and evolution, conserve, protect and restore biodiversity, and create benefits for the society and human welfare. And don't forget that genomic information is also a prior requirement for genomic editing, which is considered to be a powerful tool for bioeconomy and was awarded Nobel Prize last year, as most of you know. So what is being planned is that out of about 1.5 million eukaryotic species which are there, in the first phase, one member of each eukaryotic family will be sequenced as a reference genome. And then in the second phase, one species of each genus will be sequenced, and then all the species would be sequenced. Very ambitious program, and India is still planning to join this particular international program, but this information would, of course, revolutionize our understanding of biology and potential of use of biological information for sustainable way of development. That is what is being expected. So be ready to look for the Earth Biogenome Project output in the coming years. So from here, I will go to another way. So I told you one way of diversifying the crop genomes. The other way to diversify the crop genome is related to the technique which we know as the genetically modified food. Now for this, there are drivers like agrobacterium, which can transfer the gene to the plant, and the contribution was made by von Montagu, Mary Dale Chilton, and Robert Fraley, who were given World Food Prize in 2013, about 30 years after their discovery of this method of transforming or genetically modifying the plants. Now, this technology has been used with large number of crops like maize, soybean, cotton, canola, sugar beet, alfalfa, squash, papaya, eggplant, pineapple, and poplar. And up to now, about 585 million hectares of land is being cultivated with the technology in which the technology of gene modification has been utilized. Now remember that India has only about 150 million hectares of arable land. So a sum of land larger than what India is cultivating is being used to cultivate crops which are genetically modified. And you would be happy to know or surprised to know that developing nations are using more acreage in comparison to that of the developed nation. Like here, 99.6 million hectares in developing nations and 85.5 million hectares in case of the developed nation. Now this alone in 1 year contributed to 82 billion tons of extra food, and the bioeconomy value of this was $18 billion. So in 1 year, that much becomes the contribution of genetic modification technology, even when we have half the world is still not accepting it readily. Now in India, only one of the crop which is genetically modified has been grown over the years, and this is BT cotton, as you know. This is being grown now at about 10 million hectares out of about 12 million hectares which are cultivated with cotton. And it has provided, as the data suggests, that it has provided help to 6 million farmers, 50% increase in yield, and this is protection of yield. Remember, there is no increase in the yield by insect tolerance. It is protection of loss of the yield, which is giving this extra yield. 50% reduction in spray of insecticides and 30% increase in the economy. This alone, a single trait, is supporting bioeconomy of about $10 billion in India. So that is the power of the technology. And then imagine that in various laboratories in India, we have various crops, about 10 of these crops, including brinjal, chickpea, mustard, potato, rice, sorghum, tomato, for disease resistance, for nutritional value, for abiotic stress tolerance, which are still waiting to see the light of the day. So we hope that in the coming time, the regulatory systems would become compatible with these kind of technologies, and many more fruits of the labor of the scientists will be released in a regulated manner, so that all the fears are allayed and the benefits of the technologies are taken over a period of time. Of course, new genes are also being discovered. For example, in case of insect tolerance against the white fly, a gene has been found in case of fern. Ferns don't get affected by white fly, and when it was analyzed at NBRI in Lucknow, a new protein has been found, which, if engineered suitably in the cotton, makes cotton tolerant to that, resistant to that of white fly. So new sources of the genes have also been looked into, not only the crops, but one has to go beyond, as in this case, a protein from the fern has been found to be involved in the insect resistance. Not only genes, but also the regulatory sequences are important. For example, here we are showing our work in which a promoter which was expressing a gene in seed was taken out, and the transgenics were raised, and here you can see the blue color, which is a reporter gene. The promoter confers expression of the reporter gene only in endosperm. Such kind of promoters, when combined with nutritionally important genes, could affect the seed traits. For example, we have used it to improve the size of the grain by knocking down a gene. In case of rice, as you see here, the grain weight increased about 10 to 20% in these transgenics. Also, you all must be available of the aware of the golden rice technology. That technology also made use of a seed-specific promoter to express the genes which were already present in rice. That is another thing, that they were taken from other organisms, but rice has got those genes. They were not expressing in seeds, so they were made to express in seed. And lo and behold, we have the golden rice, which is able to express the carotenoids, which are precursors of provitamin A. And I don't have to overemphasize the utility of such a trait, because by using this, we can impact a large number of the people who have vitamin A deficiency in case of India. Another gene promoter, which was expressing in case of anther, has been combined with that of a gene for L-ornithinase. This is our collaborative work with Osmania University, with Dr. K.V. Rao. And by taking our promoter with this gene, he has been able to raise the transgenics of rice. And here you see that transgenics do not produce fertile pollen grains, whereas the female parts are not affected at all if these plants are sprayed with a herbicide, that is N-acetyl PPT, phosphothiazine, which is converted into phosphothiazine, which is a herbicide and kills the cells. So a technology for producing male sterile rice plants has been developed, and such kind of technology could be of utility for producing heterosis derived benefit to the people. Similar technology has been used in our university to produce hybrid mustard. Of course, it has got a transgenic component and it has not yet been released after regulatory clearances, but it seems to have all those benefits which a hybrid vigor trait handling could provide. So the combination of genes with that of the regulatory sequences could provide you such kind of genetically modified crops, which would be of great utility for producing more yield on less area of the land, and thus help in going towards the sustainability. Also, we isolated a gene which was showing expression under cold water and water deficit stress, mechanical wounding, and submergence. And this gene we have taken and later on identified that there are 18 genes in this gene family. So one of the gene we used and transformed into rice, and we were able to get water deficit tolerant rice. Here you can see rolling of the leaf, which is one of the trait of rice. During water deficit, leaves start to roll, but in case of transgenics, much less rolling of the leaf was observed. At the same time, more of the water was retained in the transgenic leaves in comparison to the wild type under stress condition, and stress was not only a little but sometimes it was as much low as that of 10% of the soil moisture. So these plants were able to sustain and recover when water was given to them. Similarly, we made use of a bacterial gene to transform rice, and this was evaluated at University of Agricultural Sciences in Bangalore, and we were able to show that water deficit tolerance can be conferred by genetic modification in case of rice. There is another laboratory at NIPGR which has used a RNA-based technology to silence some of the genes which are involved in ripening of the fruits. And here you see tomato and capsicum, which have been engineered with RNAi of the mannose related gene or hexaminosidase gene, and all these have resulted in increasing the shelf life. That means these vegetables can remain green for longer period or can remain healthy for longer period on shelf. So that is also possible. From this technology, that is the second technology of genetic modification, transgenics, I have told you. The first I told you about the genome assisted breeding. I would now move to the third technology, which is the technology of the day. And this is the genomic editing technology, which is known as CRISPR-Cas9 technology. For this, two women scientists got Nobel Prize last year, Emmanuel and Jennifer, and this technology based on their observation, which happens in the natural system. The bacteria which are infected by viruses sometimes acquire those sequences in their genome, and they become what we call as CRISPR loci. Now, in addition to this, bacterial genome has got a nuclease called as Cas9 and also a tracker RNA. Now this, when new virus infects this bacteria, the tracker RNA is produced and also the primary transcript from CRISPR region is produced. Now this leads to the CRISPR RNA biogenesis, in which tracker RNA recognizes these sequences from the CRISPR loci. And in addition to that, there are sequences from the infecting virus, which are there. So we have a product in which there is a virus complementary sequence, in which we have the CRRNA portion, and we also have the tracker RNA. This is able to attract Cas9, that is the nuclease, and the complementary sequence helps reach the target, and that target is cleaved by this nuclease, and therefore the infecting virus is inactivated. Now, same thing could be reconstructed, in which you have in a construct the tracker RNA, you have Cas9, and you have CRISPR array, which you can create artificially against any gene which is unique in the genome. And then, of course, what will happen is same thing. Tracker RNA would be there, CRISPR array product would be there. There would be hybridization in this. Cas9 will combine with this, and then Cas9 would be able to cut this DNA. And this DNA, of course, by non-homologous recombination system of the organism will join together, and when joining, it will cause some of the disruptions in the sequence. So you have edited in this. If you take homology to both the ends where you are making the cut, then you can integrate the whole new gene at that place, and therefore replacement of the gene would also be possible. Now, this technology has been deployed in case of rice for different genes which were found to represent QTLs for grain number per panicle, erect panicle, pran height, and grain number and grain size. And it has been possible by editing these genes and creating a mutated version that how these traits can be impacted. That is, grain number increases, here also grain number increases, and here grain size increases in these kind of variety. Now, the idea is that in this case, you do not have a foreign sequence inside the genome. Rather, what is there is a natural gene which has been muted, and therefore either it does not require regulation, as in case of transgenics, or requires limited regulation. We have to develop a clarity on that point so that farmers can get benefit of this technology. This is a powerful technology, and India is already using this technology. For example, a drought and salinity tolerant gene mutant was found and was shown to relate with the tolerance against these traits. Now, in case of this particular gene in Indian variety, this has been mutated by Chinnaswamy from IARI by using the CRISPR-Cas technology, and ultimately they were able to get the plants which are better in case of water loss and also in the salt conditions. So this technology is entering the Indian scenario as well, and should prove to be a powerful technology. To demonstrate the power of this technology, I will take this example from maize, in which case there are 6 alleles for MLO gene, and this allele, if it is mutated version, then it confers resistance. If it is non-mutated version, then maize is susceptible to this powdery mildew disease. Now, question is, if there are 6 alleles, even if a single allele is functional or non-mutated, then it will be able to confer the susceptibility. And therefore, all 6 alleles were mutated, and then these plants or edited, and then these plants became resistant. So you can edit multiple genes or multiple alleles in case of the crops by using this particular technology, that is genome editing technology. So my friends, I have started with the idea of diversifying crop genome, and I have taken 3 modern technologies which are leading the way for diversifying crop genome, and with that improving the crops for utility by our farmers and benefiting all the human race. And these three technologies has been, of course, genomics technologies, the transgenics technology, and the genome editing technology. And combination of these technologies together, either all of them or in different combination, would certainly provide a large variability in the crop genome with improved traits in coming years. That is my belief. And naturally, when we will have this situation, then we would have a scenario when we have the diversified genome assisted improvement of the crop plants, and those will be growing in larger and larger areas of the land and would be benefiting the the, the community as a whole. So that is all what I have to say today. Thank you very much once again. Hello, are you able to hear me?
Dr. Alim Khan. Hello? Dr. Alim Khan.
Sir.
So, time for questions.
Participants, please, you can interact with sir.
There is no hesitation. I know students are there. You can ask question. It at the most I will not be able to answer. That's the only possible thing. So don't worry about that. You ask your questions.
Dr. Alim Khan.
Sir.
No voice is coming from the participants.
Yeah, I have made them, they can unmute themselves if they can ask the questions. Previously, okay. I was keeping the meeting log, but I have kept it now open for participants. At okay. least, please, if you can ask any, when it's a raise the hand, I can unmute, or already it is made unmute. Sir, one question actually. So this improving the shelf life of fruits or such kind of consequences, whether it will be increasing the price of that particular fruit, or it will be because nowadays what is happening in unseason, there some fruits are available.
Right.
Maybe that is genetically modified, or some kind of modification has done. But what will be the price? Price.
Now tell me one thing. Tell me one thing. That in case of first thing, that these fruits are not transgenic, because only cotton has been released in India. No other crop has been released in India under the regulated system, and therefore the fruits should not be transgenic. The second thing is that in case of cotton, have you found cost increase because of the transgenic nature? You see, costs are sum of several things. So sometimes, and also the market, as it responds to the availability or non-availability of the commodity. So in case of cotton, I don't think that transgenic nature has increased the cost, because farmers started getting about 20-30% more benefit. So if they get more benefit, It is because they are not using herbicides. So there will be saving on that side. Whereas they will give some amount for the seed which they are purchasing to cultivate. But ultimately, the net effect is that there is 30% benefit to the farmer. And therefore, farmer would not like to make the commodity that costly that people are not able to consume it. So that is where is the market force. So what we can say is that ultimately for these also there would be initially some cost of the product, that is the technology which has been produced, but market will determine the cost and not the product. But for sure, if an industry is investing some amount into developing a technology, they would like to earn. So that much cost might be there, but market forces would also be equally operative. That's my answer.
It is a commercial crop you are talking. Okay, genetically modified crop is not increasing the price in the market. But when we are edible fruits, especially we take mango. So probably since the lockdown was stopped, we have seen that mangoes were available and some different fruits from various countries were there in the market. And especially I can quote that the mango price was near about 300 rupees to 400 rupees per kg also. So the season of mango yet to start, but it was there in market. So it was, I mean to say, it may not be means the flowering as per the we are
no. Issue is not that. In India, we do not have mango production as yet, but we are getting mango from south, because in south the production of mango starts earlier than the north. Similarly, a country might be giving, or we might be importing mango from other countries in off season, because some people are able to give high price. So that price would be a off season price. When you will go to the season of the mangoes in India, then obviously the price would not hold if it is high, because other mangoes will be available and nobody will purchase it. So it is a off-season price which you are talking about. That is on one side. And the second thing is that they are transgenic nature. No, because government of India has not approved any other transgenic import in this manner. So I would not be able to say that they could be transgenic. I don't think they would be transgenic.
Yeah,
We have a surveillance system also.
Yeah, correct answer. That in the, I mean, say, as per the climate in different countries might be different, and that may be happening that the fruits are available in that country and they are being brought to India, right? And
Dr. Alim Khan, are there any participants asking for?
We have participants, but I don't know why participants are not asking any question.
Oh, okay, no problem.
Morning we had some people ask question.
So ask them to ask whatsoever question they have. You see, it is not necessary. Sometimes we may have discussion on related themes also. It is not necessary they have to ask a specific question for a slide I have presented.
So if there are no questions, sir, Professor Akhilesh Tyagi, sir, you are the Dean of Plant Molecular Biology in India. You have made India proud by joining the global RISE genome sequencing project. And your research contributions are so immense that in the year by 2050, India's population is going to be doubled almost, and it will become number one. And in that case, though we have got 300 million tons of cereals and others stored and all that productivity is high, but nevertheless, we need food security, nutrition security, environmental security. Your lecture that has been delivered has given us a lot of information broadly on crop plant genomics and especially on genome resources and genome evolution, and then RISE genome clones and genome assisted breeding, and then all other aspects of gene pyramiding and traits prospecting, and also the disease resistance varieties and seed profile, wherein the genes contributed a lot, and also gene editing and transgenics and so on and so forth. It has been a lecture of such a kind of phylogeny and evolution dealing with biology and as well as the crop improvement. And you have given us a broad spectrum of the Indian scenario and also world scenario on this cropland genomics. And your lecture has been exhaustive, and it has been innovative, knowledgeable, and valuable lecture that you have delivered, which will be useful to our participants. On behalf of the academies, on behalf of Dr. Alim Khan, and on behalf of Telangana University, on behalf of refresher course participants, my regards to you, sir, and we acknowledge gratefully for your lecture. And thank you very much, sir, and we look forward of having fewer lectures in the years to come. Thank you very much. Thank you. Thank you.
It is my pleasure, and all the success to your program. That is what I wish. Thank you very much. You allow me to now leave?
Yes, yes, sir. Thank you, sir. Thank you very much, sir.
Thank you. Thank you very much.
Wish you good health and long life and prosperity, sir.
Thank you to you also, sir. We look forward to meet you sometime in near future.
Yes, sir. Thank you. Thank you, sir.
Thank you, sir. Thank you very much. Tyagi, sir, thank you so much.
Thank you.
Sir, with kind permission of Chari, sir, I will now end the meeting for participants also. Dear participants, please join us in time for post-lunch session. We'll be again joining back at 1:15. The lecture will begin at sharp 1:30. Kindly, all of you, please join back, and please, if you have any queries or questions, please ask at least one question. There are more than 20 participants in the participant list. Ask at least one question from one participant, please. It will make all the speakers are repeatedly reminding that you should post one question. Please, you post questions, get ready for questions in afternoon with the speaker. So in afternoon, we have two more lectures, as you know. One is from Tapan Kumar Adhyay, sir. He will be talking about soil biodiversity, life beneath our feet. And other is our S.R. Yadav, sir, from Shivaji University, Kolhapur. He will be talking about plant wealth of Western Ghats and its conservation, part 1. So,
Dr. Alim Khan, you tell the participants that if they don't listen to the lectures, their attendance certificate will not be given. Because see, why they are getting reduced at this number, you just tell them they have to come. They have to listen to the lectures. They have to ask some questions, whatever relevant is there. You please tell them, ask them to come, welcome them. Okay.
Yeah, please, please participants, pose the questions, and please make this interactive session very interesting. So hope everybody has enjoyed both lectures. Afternoon also we will be having very interesting lectures. Like both the people are very towering personalities in their own areas. You will enjoy soil biodiversity, as well as you will also enjoy S.R. Yadav sir's talk. Will be very beautifully picturizing you the Western Ghats plant wealth. So kindly join us back again at 1:15. Thank you, and have your lunch and get back to us. Thank you, sir.
Thank you. Congratulations, Dr. Alim Khan, for a very wonderful lecture.
Thank you, sir. Thank you.
You have arranged it. Okay.